At this point, we're going to talk about translation. Remember that the central dogma of biology states that everything starts out in terms of the phenotype of an organism as DNA, that is transcribed into RNA, and then that RNA is translated into a protein. And it's the protein that actually brings about the phenotype of the organism, whether that be the size of the organism or the color of the organism or the texture of the organism's external covering. All of those things are coded for by DNA, and then their actual form is brought about by the proteins. And so how do we form proteins then? Proteins are formed from RNA via this process of translation. Translation occurs in the cytoplasm. Remember that transcription occurred in the nucleus because the DNA is found in the nucleus. The RNA transcripts produced in transcription move outside the nucleus into the cytoplasm, and this is where translation is going to occur. Translation requires messenger RNA. Remember that the RNA that was formed in the process of transcription is referred to as messenger RNA or mRNA. There are other types of RNA, and we will talk about some of those even during this little lecture. Amino acids are required. This would be common sense given the fact that proteins are long chains of amino acids. Energy is needed, as you would imagine, and there are some helper molecules as well that are needed. And so we want to introduce the helper molecules at this point. Ribosomes and transfer RNA are going to be the helper molecules that play a role in this process. Now at the bottom right hand side of this screen we have the two parts of a ribosome. Ribosomes are composed of both RNA and protein. The type of RNA that we find in ribosomes is referred to as rRNA or ribosomal RNA. That's not something you find on your screen, but you can make a note to yourself that ribosomal RNA is one type of RNA that's only found in a ribosome. And so ribosomes are made of two subunits, a large subunit that we see on top and a small subunit that we see on the bottom in this diagram, that are going to come together to form the functional ribosome that actually makes the proteins. Now here's transfer RNA. Transfer RNA is the other helper molecule that plays a role in this process. And transfer RNA is a very interesting molecule. It's the third type of RNA that we have discussed. So we talked about messenger RNA, and you're familiar with the role that it plays. We talked about ribosomal RNA just now that makes up the ribosomes. And now we have transfer RNA. And so transfer RNA is depicted as the yellow molecule on the screen in front of you. It's not the orange RNA at the bottom. That's, of course, messenger RNA, a different molecule. And there's an amino acid at the top, but in the middle, the yellow portion is the transfer RNA. Each piece of transfer RNA has two different sites that make up part of its structure. The site closest to the messenger RNA in this particular diagram is referred to as the anticodon. An anticodon recognizes a particular portion of messenger RNA and binds to that messenger RNA. The other side of the transfer RNA molecule contains an amino acid binding site, and so each piece of transfer RNA binds to a particular amino acid, and depending on which anticodon that piece of transfer RNA carries, the transfer RNA will bind to a particular site in the messenger RNA, a particular sequence, a particular codon that is, and deliver an amino acid that's appropriate for that codon. So hopefully that'll become more clear in a moment, but for the time being, just remember that transfer RNA has two sites. On one end it carries an amino acid, and on the other end it binds to messenger RNA. Here's the process of translation in a nutshell. We have amino acids that are floating around in the cytoplasm, and then we have transfer RNA molecules that are also floating around in the cytoplasm. Enzymes allow these amino acids to bind to the amino acid binding site of the transfer RNA, as we can see illustrated in our diagram here. And here's where things really start to happen. So again, in this diagram, and I'll start to point now, we have the amino acids, we have the piece of transfer RNA, and once the amino acid is bound to the appropriate piece of transfer RNA, then that bound complex is going to move to the ribosome. Once it moves to the ribosome, then something's going to happen here. So the ribosome is the large purple structure here. The ribosome consists of those two subunits we mentioned previously, a large subunit on top and a small subunit on the bottom. Now fascinatingly, the ribosome has messenger RNA, this orange molecule, that's feeding through it. It's moving in this direction, the direction in which I'm pointing to here. So it's moving to the left. And what's happening is that these pieces of transfer RNA with amino acids are entering the ribosome, and they're binding to a particular site in the on the messenger RNA inside the ribosome. And so there's two things happening at different ends of the transfer RNA. On one end of the transfer RNA, then the anticodons of the transfer RNA are recognizing codons in the messenger RNA. See that? So in this particular case, the piece of transfer RNA that I'm pointing to now has a codon that says AAA, adenine, adenine, adenine. And it recognizes a particular site that's complementary found in the messenger RNA. And so that complementary sequence, of course, would be uracil, uracil, uracil. This goes back to the idea of complementary base pairing. You realize that A always pairs with U when we're talking about RNA. Now in DNA, A paired with T, but in RNA, A always pairs with U. So on one end of these transfer RNA molecules, that's what's happening. The transfer RNA molecules are binding to the messenger RNA. On the other end of these transfer RNA molecules, the protein is actually being formed. And so this transfer RNA that bound to this particular sequence in the messenger RNA carries a very specific amino acid, phenylalanine in this particular case. And so that amino acid is introduced into the growing protein at that particular point. So two things are taking place at the same time here. The recognition sites of the transfer RNA are binding to the messenger RNA, and then the protein is being formed at the other end of the ribosome. And once the piece of transfer RNA has delivered its amino acid, it's released from the ribosome, and then it goes and it's reused. And so two things are feeding out this side, the left-hand side of the ribosome. The completed chain is feeding out this side, and then the messenger RNA that's already has already been read is feeding out this side of the ribosome. And so if you understand that, and you may not after that explanation, but we're going to go back over it in an animation, which I think is very helpful, then you understand translation. That is translation. That's what's occurring. Remember, this is very small, so there are many ribosomes in a cell. So in one cell of your body that is almost, it's very, very difficult to see. Without a very powerful light microscope, you would not be able to see the cell. Then this may be happening in hundreds or thousands of instances in one single cell. Now, your body is incredibly complex, and this is what's keeping you alive at this particular point. There are thousands of proteins that have to be made continually, thousands of different types of proteins that have to be made continually in your body. And the only way that proteins are made is via translation, what we just saw here. Alright, and so this slide just wraps up our discussion there. We see this process of translation continuing. The process is going to continue until we get to this stop codon that we see here. And so the stop codon tells the ribosome that, okay, this protein that you're forming is complete. At this particular point, we're going to cut off the protein and release this protein. And so here we see that occurring. The stop codon, the ribosome read the messenger RNA all the way to the stop codon. Once that stop codon shows up inside the ribosome, the protein is released and goes on to the rest of its final destination. It may go through a few steps in the meantime. It may go through the Golgi complex. You may remember that from earlier in our class. We talked about how in the Golgi complex proteins are formed into their final shape and then transmitted to wherever it is that they're going to reside in the cell. That might be inside the cytoplasm, that might be inside the actual plasma membrane. If it's a plant cell, it might be the cell wall, and so forth. Alright, and so what we want to do at this point is to take a look at an animation that illustrates this process. Oftentimes I feel that an animation helps more than anything else in tying together the understanding of a process such as translation. And so let's take a look at this animation. This is the one I always use in class to illustrate this process. What we see on the screen then are the two parts of the ribosome that you will recognize, the small subunit and the large subunit. It's interesting to note that every time a protein is synthesized, these two subunits are floating around in the cytoplasm and they assemble onto the RNA. They don't remain in the cytoplasm assembled and then go look for messenger RNA molecules but they assemble onto the RNA each time a protein is produced. And so that's what's going to occur in this particular case. In this case, I'm less concerned that you're familiar with these various sites. There are several sites that are located in the ribosome. So here we have the piece of transfer RNA, so you do know all about these sites. So here's the transfer RNA. On one end of the transfer RNA we have an amino acid. On the other end of the transfer RNA, of course, we have our anticodon. Alright, let's move on a bit. And we'll let the video play on. Okay, so there is our messenger RNA. And AUG is going to be important to us in the next lecture that's part of this chapter. AUG is a start codon, and so that's what tells the ribosome to start synthesizing a protein. So here's the large subunit sliding along the messenger RNA until it gets to the start codon. There is that first amino acid that binds the anticodon here, binds to the codon there in the messenger RNA. The small subunit, the large subunit, the small subunit's there, the large subunit comes along and binds to the messenger RNA there. So it was the small subunit that was there initially. Here comes another piece of transfer RNA, and it binds, as you can see there, also to its complement. So I think we all understand this at this point, but this is the anticodon on the transfer RNA, and this is the codon found in the messenger RNA. You remember that a codon is a sequence of 3 bases. You know, a codon may be a term that I have thrown in a little bit early. We're going to talk about it in the next lecture. But if you will just take my word for the time being that a codon is simply a series of 3 bases found in a nucleic acid, RNA in this case, then we'll be in good shape as far as that goes. And so the anticodon of the transfer RNA binds to the codon in the messenger RNA, and now exciting things start to happen. Here we see a bond that's formed between the 2 amino acids that have been introduced by these pieces of transfer RNA. This bond is referred to as a peptide bond, a term that will be familiar to you from the early portions of our class. And interestingly, what's going to happen now is that the piece of transfer RNA that delivered that first amino acid is going to leave. Everything scoots over one spot inside the ribosome, and here comes another piece of transfer RNA. That piece of transfer RNA binds to the messenger RNA in a complementary fashion, anticodon to codon. Another peptide bond is going to be formed, and then that piece of transfer RNA leaves. And so this is going to occur over and over and over again. Remember that most proteins are typically 1,000 or thousands of amino acids in length, and so this is going to take place. It takes place incredibly rapidly. I mean, hundreds of these bonds are formed within a matter of a second, which is incredible and difficult to comprehend, but it takes place very, very quickly. Now, what's being illustrated here, we're not going to do 1,000 in this little animation. We just do 3. And here the stop codon is encountered. And so we'll talk about stop codons again in the next little lecture in this chapter. But remember that a stop codon says to the ribosome, okay, this protein is done. You need to let this protein go and stop the process of translation. And so the release factor binds to the stop codon found right here. And the release factor does not specify an amino acid. It does not bring an amino acid to this growing protein. So now the protein is gone, and interestingly, lastly here, everything is going to disassemble. There goes the last piece of transfer RNA, there goes the release factor, and there goes the 2 parts of the ribosome. And so that is the process of translation.